Natural IgE monoclonal antibody defines a unique epitope on DERP2. This study was performed by Indoor Biotechnologies in collaboration with Dr. Max Cruz from the University of South Carolina for the crystallographic studies and with Dr. Scott Smith from Vanderbilt University Medical Center for the isolation of human IgE monoclonal antibodies. The rationale for this study is there is a limited knowledge of the epitopes associated with the human IgE repertoire. The main reasons for this limitation are the challenge of one, cloning IgE antibodies because of the extremely rare B cells harboring IgE genetic information, and two, mapping monoclonal antibodies on the allergen. Our aim was to identify and analyze epitopes recognized by human IgE monoclonal antibodies as they occur in vivo for subsequent design of hypoallergens for immunotherapy. The first step to make human IgE producing hybridomas was the isolation of PBMCs from blood of an allergic patient. B cells were grown in 96 well tissue culture plates for 7 days before primary screening by ELISA. Cells within wells containing IgE were then fused to a myeloma partner by electrical cytofusion. Human hybridomas were then selected in hot medium and biologically cloned using flow cytometric single-cell sorting. Finally, IgE secreting hybridomas were grown in serum-free medium before IgE monoclonal antibodies were purified by affinity chromatography. Using this human hybridoma technology, 4 IgE monoclonal antibodies against DERP2 were isolated from 2 allergic subjects. DERP2 was selected as the model system because of its relevance as a major allergen from the house dust mite. In the study shown here, we used NMR to map epitopes on the allergen. On the left, you can see a unique epitope that was identified for 2F10 IgE monoclonal antibody, shown in purple, that is far from the area where the other 3 IgE monoclonal antibodies bound. On the right, relative epitope mapping was assessed by immunoassay. 3 of the 4 IgE monoclonal antibodies did not bind to DERP2 presented by a murine IgG monoclonal antibody alpha DPX. This indicates that their epitopes overlapped. In contrast, the epitope for the IgE monoclonal antibody 2F10 clearly bound to a different site, as also shown by the NMR data. The epitope for this IgE antibody 2F10 was further investigated by X-ray crystallography and site-directed mutagenesis analysis. Recombinant 2F10 FAB was expressed in mammalian cells. DERP2-0103 was expressed in Pichia pastoris. The X-ray crystal structure of the allergen in complex with the FAB was determined at 2.2 angstrom resolution by molecular replacement. This unique epitope, shown in purple, interacted with the antibody through hydrophobic interactions and hydrogen bonds. Antibody inhibition assays showed that mutation of 2 amino acids, aspartate and leucine, involved in hydrogen bonds led to a reduction of either 10 or up to 1,000 times reduction of IgE antibody binding, depending on the substitution. Similar strong reductions of up to 1,000 times were obtained by other substitutions of an adjacent epitope residues or lysine involved in hydrophobic interactions. These results indicate the importance of knowing the specific interactions and residues involved in the allergen antibody recognition for a successful reduction of IgE antibody binding. In conclusion, we report, to the best of our knowledge, the first X-ray crystal structure of an allergen, DERP2, in complex with a FAB from a human IgE that has the natural pairing of heavy and light chains. Epitope analysis will facilitate the design of hypoallergens for immunotherapy and contribute to understand the IgE antibody repertoire. I finally want to express thanks to NIEAD from NIH for funding for the NIH Award Arrow 1 to Indoor Biotechnologies that funded this research. Thank you very much.